I head the small plant molecular biology group at the Instituto Gulbenkian Ciência near Lisbon, where we use Arabidopsis as a model system and have been focusing on two major lines of work. The first aims at uncovering the functional, the biological relevance of alternative splicing in plant systems, while another subgroup of lab members is looking into the physiological roles of a particular class of membrane transporters called the major facilitator superfamily, or the MFS. And what has been happening is that these two projects have become tightly intertwined as our analysis of these transporters has been revealing striking examples of the biological impact of alternative splicing. Okay, so together with the ABCs, the MFS represents one of the two largest groups of transporters on Earth, and these are also the only two groups of transporters that are known to occur ubiquitously in all classes of living organisms. So while I'm sure most of you have at least heard of ABC transporters, the MFS remains very poorly characterized, and many of you might not even recognize the name. So unlike ABCs, which hydrolyze ATP for their activity and are in general multi-component transporters, MFSs are always single polypeptide carriers that transport small molecules across membranes using electrochemical gradients as energy source. The characteristic MFS protein structure consists of two transmembrane domains with 6 membrane-spanning segments each that flank a central hydrophilic loop. The Arabidopsis genome has more than 120 genes predicted to code for putative MFS carriers, and the very few that have been characterized to date have been implicated in sugar, nitrate, or phosphate transport. Apart from these 3 small clusters, one other MFS carrier called ZIF2 is involved in zinc and iron transport. And we were interested in understanding whether the MFS would play a broader role in metal homeostasis in plants. And indeed, we found an additional gene, which we later called ZIF2, involved in regulating the tolerance of the plant to zinc. Zinc is an essential micronutrient in plants. It is, for instance, known to be required as a cofactor for more than 300 enzymes, and the symptoms of zinc deficiency include leaf chlorosis and browning in many important crops. Zinc, however, is also highly toxic when present at high concentrations. Moderate zinc toxicity induces leaf chlorosis and wrinkling, whereas in heavily contaminated soils, such as here near a zinc mine, the landscape can be devastating. So to cope with these opposing effects, plants have developed a tightly controlled array of mechanisms to maintain zinc homeostasis. In the case of high zinc, they respond by reducing the uptake or actively extruding the metal ion from the root system. And there are also some tolerant hyperaccumulator species which are able to accumulate large amounts of the metal in the above ground parts of the plant. Similarly, at the cellular level, zinc tolerance is achieved by reduced uptake or increased efflux, and it can also be sustained by the vacuolar sequestration either of free zinc ions or upon conjugation with specific ligands. So clearly, all these steps require the action, the concerted action of different transporters. And as I told you, we found one gene, which we called ZIF2, whose knockout displayed a clear zinc-related phenotype. Under control conditions, under optimal exogenous zinc concentrations, 30 micromolar, the mutant is indistinguishable from the wild type. For those of you not familiar with Arabidopsis work, when you see this, it refers to the Columbia ecotype, which we used as the wild type throughout this study. However, the mutant exhibits a clear exacerbated response to zinc toxicity, as you can see here, exhibiting namely reduced primary root elongation, lower shoot biomass and chlorophyll content than the wild type. So loss of ZIF2 function results in oversensitivity to high zinc stress. To investigate the expression pattern of this gene, we then generated transgenic plants expressing both the GUS and the GFP reporter genes under the control of the endogenous ZIF2 promoter, which we found is highly active at the cortical cell layer of both primary and lateral root tips. So RT-PCR analysis confirmed that ZIF2 is most highly expressed in root tissues, and also revealed the production of 2 alternative transcripts, which in roots are expressed in an approximately 1-to-1 ratio. So sequencing of these 2 bands indicated that the 2 splice variants result from an intron retention event in the 5'UTR of the ZIF2 gene. This intron is 139 nucleotides in length. We were unable to find any alterations in the coding sequence, nor any in-frame ATGs or alternate start codons in the retained intron, indicating that both splice variants code for the same putative MFS transporter of approximately 50 kilodaltons. Okay, so we next wanted to analyze the subcellular localization, so the site of action of this transporter. So we fused the ZIF2 cDNA with YFP, transfected them into isolated protoplasts, and concluded that this transporter is localized at the membrane of the vacuole, which in plants is called the tonoplast. Stable expression in transgenic Arabidopsis root tips also confirmed tonoplastic localization of this transporter, and not surprisingly, since they're expected to code for the same protein, when instead of the shorter ZIF2-1, we cloned the full-length cDNA of the longer transcript with the retained intron in the 5'UTR, the results were identical. I have someone to check my minutes now to just keep track of my time. Okay, so co-localization experiment with both a tonoplast and a plasma membrane marker clearly revealed that co-localization of our MFS carrier is only observed with the tonoplast marker. So ZIF2 is localized in roots at the cortical cell layer and at the tonoplast, at the vacuolar membrane of the cells. Okay, so we then used heterologous expression in yeast to study the transport activity of this carrier. And in normal media containing 2.5 micromolar of zinc, wild-type Cerevisia cells expressing the Arabidopsis ZIF2 transporter show no significant growth differences when compared to cells transformed with the empty vector. But cells expressing ZIF2, their growth is substantially less inhibited than the control cells when zinc is present in excess. So to confirm this, we used a yeast mutant that lacks 2 zinc transporters and is hypersensitive to high zinc stress. And as you can see, expression of our plant transporter in this mutant background fully rescued the yeast mutant oversensitive phenotype. As shown in this table, expression of ZIF2 reduces the content of the metal by 65 to 80%. So taken together, these results are consistent with the ZIF2 transporter functioning to extrude zinc from the yeast cell. Okay, so coming back to the plant, given our mutant phenotype and the high expression that we detected in roots, we next asked whether ZIF2 expression would be zinc responsive in root tissues. And as I showed you before, under standard zinc conditions, the ZIF2 mRNAs are expressed at approximately equivalent levels, and these splice variants expression is not affected by zinc deficiency. However, a clear shift in the splicing pattern of the gene is observed in response to even under moderate zinc toxicity, with the longer ZIF2 transcript being highly upregulated, while the shorter ZIF2 is also induced, but to a lower extent and only under high zinc toxicity. QRT-PCR confirmed that both total transcript levels of ZIF2 and that of the ZIF2 splice variant are highly induced by zinc. So both expression of the ZIF2 gene, total expression of the ZIF2 gene, and alternative splicing of its pre-mRNA appear to be regulated by the plant zinc status. Okay, so to investigate the functional significance of these two different splice variants, we generated transgenic plants overexpressing independently the two transcripts and selected 3 transgenic lines of each type for phenotypical analysis. By RT-PCR, we were able to check that all transgenic lines expressed similar transgene levels, and by the way, also that the retained intron is not spliced out from the longer ZIF2 gene. And what we found is that ZIF2 overexpression significantly attenuates the detrimental effects of zinc toxicity, so the opposite phenotype that we found in the mutant. And importantly, we noticed that the longer ZIF2 splice variant consistently induced a more pronounced effect than the shorter ZIF2. You can see here, upon quantification of shoot biomass, chlorophyll content, and root growth, that there is a range of zinc oversupplies where the longer ZIF2 confers about twice the tolerance than ZIF2. So the longer transcript that harbors the retained intron, which I just showed you is preferentially expressed under high zinc stress, also contributes more significantly to plant zinc tolerance. Okay, and to gain insight into how ZIF2 might be functioning in the response to zinc toxicity, we then quantified by atomic emission spectroscopy the endogenous plant zinc levels in our different genotypes. And we first found that at the whole plant level, the total amounts of the metal are unchanged in wild-type, mutant, and overexpressing lines, indicating that ZIF2 is not conferring tolerance by reducing uptake or actively pumping out the metal from the root system, which was anyway not consistent with the fact that our transporter is not at the plasma membrane. But when we quantified zinc independently in the shoot, so the aerial part of the plant, and the root, we found a different picture. So the above ground parts of the mutant accumulate more zinc than the wild type, while the overexpressor accumulates less. Whereas in roots, the opposite trend is seen. The mutant contains less zinc and the overexpressor is more zinc than the wild type. So basically, in the mutant, the zinc content is biased towards the shoot, whereas in the overexpressor, it is skewed to the root. Okay, sorry for that. So this experiment revealed two important things. First, that ZIF2 functions to retain zinc in the root and thereby prevents the translocation of the metal to the above parts of the plant where it can be more toxic to the plant. And secondly, in clear agreement with the physiological data I showed you in the previous slide, ZIF2, the longer transcript, also contributes more significantly to this partitioning of zinc in the plant. So since the two transcripts code for the same protein, the only explanation we could think of for this differential contribution was that the ZIF2 transgenic lines were actually producing more protein, actually contained more protein than ZIF2. And in fact, when we generated transgenic plants expressing YFP tagged versions of each splice variant, it immediately became apparent that the ZIF2 plants could be producing more protein. So we assessed by QRT-PCR the transgene transcript level, so we're looking at the YFP transcript in the transgenics, and then upon quantification of the fluorescence signal here and normalization to the corresponding mRNA levels, we found that indeed the ZIF2 plants contain about 2 to 3-fold more protein than the ZIF2 counterparts. So retention of this intron apparently induces more translation inside the plant of the ZIF2 transcript. So we next cloned independently the two 5' UTRs alone under the control of a strong promoter and upstream of the luciferase gene, and transfected these two constructs into isolated protoplasts to measure luciferase activity. And consistent with what you observed in planta, about twice the luciferase activity was observed with the longer 5' UTR than with the shorter one. And when we repeated this assay under a range of zinc conditions, we found that while the shorter 5' UTR, with the shorter 5' UTR, there is not a substantial induction with zinc, translation of the construct bearing the longer 5' UTR is markedly upregulated by zinc. So retention of this intron markedly promotes protein expression in a zinc-responsive fashion. Okay, so finally what we did is secondary structure prediction of this 5' UTR revealed the formation of an extensive and stable imperfect stem loop upstream of the ATG in the ZIF2 5' UTR, which is lost in the ZIF2 because splicing out of the intron removes roughly half of the sequence participating in this structure. So to summarize what we have learned of ZIF2 so far, it is a tonoplastic transporter predominantly expressed in the cortex of root tips that acts as a plant zinc detoxifier, since we found that knockout is hypersensitive and the overexpressor tolerant to high zinc stress. Using yeast, we were also able to show that this MFS carrier functions in zinc transport, and in planta, the overexpressor we know retains more zinc in the root. So from this, we can conclude that ZIF2 sustains plant zinc tolerance by mediating the vacuolar sequestration of zinc ions in root cortical cells, and then thereby preventing its translocation to the above ground parts of the plant. On the other hand, we found that intron retention in the ZIF2 gene generates two splice variants that differ in the length of their 5' UTRs. And last but not least, zinc regulates expression of ZIF2 at multiple levels to enhance tolerance to its toxic effects. So the total steady-state transcript levels are induced by high zinc, which also shifts the alternative splicing pattern towards the production of the longer 5' UTR transcript, which then will confer more tolerance to zinc in the plant. Finally, we found that this retained intron promotes protein expression in response to zinc. So by regulating the levels of a zinc-responsive mRNA variant, alternative splicing controls ZIF2 translation to promote plant zinc tolerance. Just before I finish, I just want to say that the driving force for all the work I showed you today is a very talented postdoc from France in the lab, Stel Remy, who had the important help of Rita Batista and the precious collaboration of Isabelle Sacoure's yeast group at Instituto Superior Técnico, and also the help of my colleague at the IGC, Alekus, who actually told me last week that he worked here for some time at the ICGEB and actually asked me to check on Franco Pagani. So thank you very much for your attention.